I'm Uduak Thomas, Senior Editor with Jen, and it's my pleasure to be your moderator for today's webinar. Our speaker today is Dr. Erik Johansen, and he is an R&D application scientist at CDI Labs. Before we get started with his presentation, let's get a bit of background on today's topic. Membrane proteins are key targets for antibody-based biologics. In fact, over 60% of FDA-approved drugs act on these proteins. Studying them is challenging because the proteins have to be embedded in lipid bilayers to retain their native structure. Many membrane proteins also require post-translational modifications such as glycosylation to function properly. Current technologies for studying membrane proteins either involve expression in cell-based systems, which can lead to high background from contaminating endogenous proteins, or purification using detergents or nanodiscs. In both cases, these approaches can result in receptor inactivation. So in this webinar, our speaker will demonstrate the utility of virion-displayed membrane proteins for screening antibody biologics. He will present the VirD membrane protein array technology, which displays membrane proteins in the membrane envelope of herpes simplex virus 1 or HSV1 virions. The technology is designed for high-throughput biochemical analysis of membrane proteins. Membrane protein expression occurs during virus replication, ensuring that the integrated proteins undergo endogenous folding and post-translational modifications in their native and active conformations. He will also share the results of testing various commercial and clinical antibodies.
Thank you for the introduction. I'm happy to be talking to the audience today more about the virion-displayed technology and how we can use it as a highly multiplexed analysis to look at different types of biologics. I'd like to begin with some background on G-protein coupled receptors, otherwise known as GPCRs. Basically, what GPCRs do is that they transmit information on the exterior of the cell to the interior. They do this by binding a ligand and signaling through G-proteins. How this works is when a GPCR binds a ligand, it causes the transfer of the inactive GD-bound state of the G-alpha protein to GTP bound. What this does is it then activates it, it dissociates from the beta-gamma subunits, it stimulates an effector protein, which can then go on to induce a response within the cell. There are many different types of GPCRs expressed in many different cell types. And depending on the context of the cell, the ligand, and the effector proteins and the G proteins themselves can cause a wide array of different types of signaling pathways, which is what makes them very attractive for treatment of disease. Over 34% of all FDA-approved drugs target GPCRs. As such, this makes them one of the most heavily targeted receptor families amongst all membrane proteins. Looking at the total number of GPCRs, being 398 non-odorant GPCRs, 27% of them have established FDA-approved drug targets, otherwise known as 108 receptors. These primarily act on opioid and aminergic receptors. Of those entering clinical trials, these are being developed against GPCRs that are of the peptide class. I'm sure one thing that we are familiar with is that the recent success of semaglutide, otherwise known as Ozempic, has seen massive success both therapeutically and commercially. So there is a need and a desire for new drugs to be made against GPCRs. The fact remains, though, that over 50% of all non-odorant GPCRs have yet to be targeted by any small molecules or biologics. And this, in part, is due to the inherent difficulties of studying multipass transmembrane proteins. Part of the difficulty of studying multipass transmembrane proteins is that they require being embedded in a phospholipid bilayer. This is necessary in order for them to retain conformation to bind their endogenous ligands. As such, one of the most immediate solutions is to do the screening in cells. However, many of the times, overexpressing these GPCRs in cell lines can be toxic. As such, another solution would be to express them heterologously in either E. coli or yeast. But these proteins require specific conformations and post-translational modifications in order to function. As such, it is difficult to recapitulate these types of modifications in E. coli and yeast. If we were then to use cell types and try to purify them, this too also has its own share of difficulties in that the use of detergents often cause delipidation of these receptors, which can cause them to lose their conformation and inactivate them. Furthermore, if we do a high functional screening or high throughput screening of drugs via functional assays, this can be difficult for receptors with no known ligands because we need knowledge of which G proteins they signal through in order to observe an effect. So there is a need for an unbiased approach towards ligand binding with the receptor. One such solution to these issues is virion-display to study GPCRs. Virion-display is a technology by which we replace a non-essential glycoprotein, in this case glycoprotein B, with that of a human multipass transmembrane protein. To date, we have done it the most with GPCRs, but it is amenable to any type of membrane protein. And in doing so, herpes simplex virus 1 is an enveloped virus particle, and when we replace this glycoprotein with that of a GPCR, it remains embedded in the lipid bilayer. We choose HSV1 for its specific capability in being able to hijack the secretory network of host cells. When the virus is replicating, it is going to shut down endogenous cell protein expression and prioritize the expression of all viral proteins. And when we genomically modify the genome of HSV1 with the GPCR, we are replacing the control of the GPCR under viral promoters. As such, when the virus replicates, it is driving the expression of the GPCR. As the virus is being assembled, it's going to derive its lipid envelope from the Golgi apparatus. And this is important for membrane proteins because endogenously, when GPCRs are being expressed, they too will move through the secretory network in order to get decorated with many different types of post-translational modifications before ultimately being expressed at the plasma membrane. Since the virus is going to move through this same network, it will ensure that this GPCR gets all of its native post-translational modifications. The final result will be an intact virus particle with a fully decorated GPCR with all of its native PTMs while never having left a lipid envelope, and thus ensures that it's using the host cell machinery to ensure efficient and proper translation. To make these virions, as I previously mentioned, we're going to genomically modify that of HSV1 with the human GPCR. We replace the GB gene locus with our GPCR ORF, and we amend a C-terminal V5 tag in order to monitor expression of the GPCR. We choose a C-terminal tag because many GPCRs tend to undergo processing at the N-terminus, and thus it's more likely that we could lose the tag while it is moving through the secretory network. As part of the QC, when we make these virions, we're able to infect any cell type that's permissible to HSV1 virus replication. And since all of these GPCRs have a C5 terminal tag, we can analyze the expression of the GPCR via Western blot, and we can also purify the genome from the virus to make sure that the sequence at this gene locus is indeed correct. With the resulting virions, we're able to then fabricate the virion display array, in which we take purified virions for each individual GPCR in our collection and spot them onto functionalized glass slides. To date, we have done this with 291 non-odorant GPCRs. This represents roughly 70% of all non-odorant GPCRs, but we have since expanded our collection to include a series of receptor tyrosine kinases, as well as popular immune signaling targets. For this webinar, I'll be discussing a few of the applications of the virion display technology, but the one that I'll be spending the most time on is how we can use this for highly specific biologicals. In the Nature Communications paper that was published for this technology by a lab in Johns Hopkins University, they screened over 20 mouse monoclonal antibodies that are commercially available to assess the specificity and to ensure that the GPCRs incorporated on our virions are indeed correct. And the result that they found was there were only about 10 of these 20 that had specific interactions with the GPCRs. The rest had quite nonspecific binding on the array surface. And this highlights one of the more powerful uses of the technology in which we can do both positive screening against our intended GPCR target, but also counter screening against those targets that we may not think will bind, but are showing as a strong nonspecific binder. There are three types of categories that we tended to see with this type of screening. The first are those antibodies that are ultra-specific. They bind their intended target with no other cross-reaction to any other type of GPCR. The next were those that specifically bound their intended target and would be our top-ranking Z-score hit, but they had some specific off-target effects as well, as is the case with the ACR3. It bound to CalCR and GPR61. And then we had those antibodies that failed to recognize their targets altogether. But it stands to reason that there is likely that there could be a problem with our virion, and it may not be a problem with the antibody. So to demonstrate that the GPCRs are properly expressed by virions, we infect the cell line with the viruses in order to drive expression of the GPCR. In this case, we are doing a cell surface stain with a fluorescently labeled antibody, the anti-ACR3 as previously shown, to demonstrate that it is in fact binding ACR3, CalCR, and GPR61 as we saw on the array format. We know that the GPCRs all have a C-terminal V5 tag, so when we probe by Western blot the expression by V5, we can see that similar staining patterns occur with ACR3 and the V5 tag, showing that the GPCRs are in fact expressed, and this antibody is interacting with these GPCRs specifically. As is the case for DRD1, we do not see a similar pattern. DRD1 infected cells fail to be recognized by this antibody above our negative control. Our negative control is called K082. This is basically the empty virus with no human orthogonal proteins. So it'll have all the other viral glycoproteins that you'd find on HSV1. When we analyze this by Western blot, we can see that the anti-DRD1 antibody also failed to recognize the target by Western blot, but we can see via V5 tag that the protein is in fact being expressed. Then how do we know that the DRD1, as seen by V5, is in fact correct? We had the D1 antagonist and observed the binding via turf microscopy. I'll go into this in a little bit more detail later, but this basically shows that we are able to observe specific binding to these cells that are expressing the GPCR over our negative control, confirming that this GPCR is in fact properly expressed, properly folded, and can bind its endogenous ligand. As further proof of other antibodies that can recognize the ectodomain of GPCRs, we see here for the ACR1 and GPRC5C, they are both able to recognize their intended target. ACR1 is printed in 2 locations on our array, and each bind quite specifically to the antibody. For some other antibodies that we have, we see a similar pattern, but with some more background. As is the case of MERG-PRF, we still see specific binding towards the intended target, but we start to see some more noise and background with other GPCRs that are expressed on the array, suggesting that this antibody isn't as clean as the others that were tested. For SSTR2, we also see a similar pattern, as was the case with ACR3, in that the top 2 hits were against unintended targets, IGF-1R and EphA3, but it was still able to recognize its intended target of SSTR2. So while using mouse monoclonal antibodies is great for validating commercial reagents and to prove that our GPCR virions are in fact properly expressed on the surface of the array, it's far more interesting and impactful if we can run clinical antibodies that are being used therapeutically today to see just how specific these antibodies are. So we did this with 4 types of the most used therapeutics, being pembrolizumab, nivolumab, trastuzumab, and mogamulizumab. And while we are specifically talking about the virion display arrays, CDI also offers a service and product called the HuProt protein microarray. These arrays contain over 80% of the full human proteome produced as N-terminal GST fusion proteins from yeast. When we ran pembrolizumab on a combo array, being the HuProt array and the virion-display array printed on one single functionalized glass slide, we get images that look like this. For printing control, we can do an anti-GST and an anti-GD combination treatment. All of the proteins that are produced from yeast for the HuProt, and again, this has over 21,000 proteins, will have an N-terminal GST fusion. The virions have other glycoproteins that we can tag with antibodies, namely a common one is glycoprotein D. When we do this, we can see every single spot on the array light up, showing that there is in fact protein or virion printed. In order to determine where pembrolizumab is binding, we look specifically with an anti-human IgG against the FAB region. And we can see a relatively blank HuProt array. This is because pembrolizumab, amongst all 21,000 proteins printed on the array, it only binds to a single place, and that is PD1, which is rather remarkable in that out of all the proteins on these arrays, it doesn't have any cross-reactivity with anything else. So it is truly a gold standard for how specific a therapeutic antibody can be. We see something similar for nivolumab, in which it too only recognizes a single target, being PD1, across the entire HuProt collection. Down below, we can see that there is another hit here. Sometimes what will happen with these arrays is a fleck of dust will remain, cause artifacts and fluorescence, but we know that this is not a real hit since the fluorescent pattern doesn't match what the printed proteins should look like. When testing trastuzumab, we see that there is binding in 2 areas, both on our HuProt express clone, being a purified N-terminal GST protein, and that is expressed as part of the virus particles. For PD1, the only area that showed binding was on the virion display area. It is worth noting, as previously mentioned, that the virion display is meant to complement what our HuProt collection may not have properly expressed or filled, since those are using detergents in order to purify them from yeast. Virions do not require any type of purification or detergent method, so they are being presented very well endogenously. And clearly the behavior of how those extracellular domains will fold will be variable depending on the protein. But this is a good example of how we see that trastuzumab is able to bind both clones, and that they are the top 2 hits, and that no other protein amongst all printed on the array interacted with the trastuzumab. Mogamulizumab is only one of 2 FDA-approved antibodies against GPCRs, the other being talquetamab against GPRC5D. In this case, we see that it was able to bind the CCR4 in our HuProt collection. In this case, for the combo array, the signal intensity for the virion was not high enough, but when we do the VirD-only arrays, which I'll get to momentarily, it too is able to bind But again, we see the same pattern, that it is interacting with a single protein amongst all other proteins, highlighting just how specific this antibody is. As mentioned, when mogamulizumab is run on our VirD-only arrays, we're able to see that it is indeed able to bind CCR4. Now, we also see on this analysis that FcGR2a lit up as a top hit. Included on our virion-displayed collection are the Fc receptors against many different types of Ig molecules, including IgG. Fc gamma R2 and Fc gamma R1 are both high affinity receptors for the Fc region of IgG. Given that we are adding human antibodies with the Fc region for IgG, it makes sense that we would see binding to these areas. We want to ensure, though, that the binding to these areas is caused by the Fc receptor interacting with the Fc region of the IgG, and that it is not the CDR region of the antibody interacting with the receptor displayed on the surface of the virion. And we are able to do this if we treat the antibodies with enzymes that cleave away the Fc region. All of this is to say that this is not unexpected, and it does not mean that the antibody is nonspecific. It is merely that the proteins that we are expressed on the surface of the virion are doing what they do endogenously. And engagement of the Fc receptors endogenously and when used as a therapeutic is important to elicit an immune response, be it with T cells or NK cells or neutrophils. Similar to mogamulizumab, we ran the other clinical aliquots of these antibodies on our VirD-only arrays, and we can see both in nivolumab and pembrolizumab, PD-1 is the only spot to light up, and trastuzumab is the only spot to light up for HER2, otherwise known as ERBB2. We also had at our disposal a clinical aliquot of rituximab. However, this antibody is a chimeric antibody in which it has mouse CDRs with a human Fc region. Our protocols are optimized for detecting the FAB region of IgGs. As such, we couldn't use our optimized secondary antibodies, which is what caused some higher background stain with this particular array. When we analyze the data, however, the intended targets are among the top hits. We can see for rituximab that CD20 is in fact the second ranked hit, and that Fc gamma R1, that's that is the top ranking hit just because that affinity complex is quite strong. Hopefully with all of that data, I was able to convince you that the VirD arrays are in fact useful for screening antibodies just to test how specific they can be, and that these GPCRs presented as antigens are correctly folded in order to accurately determine whether or not an antibody is binding the ectodomain. These virus particles are closed substrates. The intracellular domains are not available for the antibodies to bind to, so it limits what we can see to the extracellular region. As another control to show that the GPCRs are indeed folded correctly and expressed properly, we can run peptides on the VirD arrays as well. We can see that when we add, in a rather simple setup, fluorescently labeled peptide, and it need not be a fluorescent label, it can be any other type of affinity handle that we can then add a secondary reagent with the fluorescent label in order to visualize where it's binding. When we add these types of peptides, they'll bind to the receptors, and we'll be able to correlate the strength of the binding based on the fluorescent intensity. We did this for two types of peptides, namely dynorphin A and SIRF14. Dynorphin A is a ligand, an agonist for the delta opioid receptor. SIRF14 is an agonist for SSTR2. For dynorphin A, we see what we'd expect in that OPRD1 was a top-ranking hit. However, for SIRF14, we see that while we were able to observe binding to SSTR2, we had some other hits to unexpected GPCRs, being NTSR1, kisspeptin, and GABR2. We do a similar experiment as we did with the antibodies, in which we can add the fluorescently labeled peptide to cells that are infected with the virions to drive expression of the GPCRs and quantify the relative signal intensity of these cells expressing the GPCRs. When we do this, we can see that in this top row, when we just add the fluorescently labeled peptide, we get significant signal intensity for all of these GPCRs above our negative control. We can also do a competition assay in which we'll add unlabeled peptide in combination with the labeled peptide, and this consequently drives down the signal intensity. Lastly, we can add a similar type of analog of SIRS, cyclostomatatin, and we see the same pattern which drives off the signal, or the fluorescently labeled peptide binding to the GPCRs. So this is indeed showing that the GPCRs are binding the peptide specifically and that it can be competed off in a specific fashion. Lastly, I will discuss how we can use the virion-displayed technology for label-free small molecule screening. This technology was more of an exploratory measure to see what we can do with the virions. It is not currently a service that we offer at CDI, since it requires a home-built setup with collaborators at Arizona State University. But all the same, it is an SPR-based method in how we can do label-free detection of small molecules. The way this technology works is that we can take the purified virions and use peg linkers to attach them to a gold slide. In doing so, we are able to measure the change in surface charge of the virion when we add a given analyte, in this case, small molecule. When a small molecule binds, it's going to change the overall level of surface charge of the virion. As such, when we apply an alternating electric field, this will cause the virions to oscillate. And the amplitude change of the oscillation of the virions from the gold surface will induce an SPR response, which we can measure. The higher the concentration of peptide, the larger change in surface charge there is, the larger the amplitude change will be, which is exactly what we see for these four receptors, GABR2, kisspeptin, SSTR2, and NTSR1. Whereas when we look at our negative control, regardless of the type of peptide we add, there is no amplitude change because there is no specific binding to those virions. As I mentioned earlier, we can also use turf microscopy to observe binding of the native ligands to the GPCRs. Turf microscopy is a method by which you can look at a very thin layer of a given cell, or in this case, virus particle, and only look at the membrane layer in which the GPCR is contained. When we do this with fluorescently labeled ligand, we're able to see that each GPCR is able to bind its cognate ligand specifically over our negative control. K-delta UL27 is synonymous with K082. These are just the empty vector HSV1 virus particles. We can see for ACR3, there is specific binding to its ligand CXCL11, CCR1 with CCL23, apelin with its receptor, and CXCL1 with CXCR6. In each of these cases, we see the same thing in which there is specific binding to the GPCR over our negative control, further demonstrating that these GPCRs are in fact expressed, properly folded, properly post-translationally modified in order to bind their cognate ligands. Here at CDI, we are always looking on ways to expand the applications of our virion-displayed technology. I've gone through how we've done it with highly specific biologicals and how we've done it with peptide screening. We can also use these virions to do GPCR deorphanization. This is what my thesis project was at Hopkins and is entering the review process, so keep your eyes peeled for that. But we can still use this to do drug screening, Aptamers are becoming more prominent as a new class of therapeutics, as well as looking at pathogen GPCR interactions. So we encourage you to think of how these virions could be a tool for your future research. And on that note, the virion-displayed technology that preserves the native protein conformation and modification of these membrane proteins. They are available for purchase as a microarray for your own use, or we can do it as a service here at CDI. There's special pricing available for combining our HuProt arrays and our VirD arrays. If you are interested in using the purified virions as a reagent tool, we encourage you to contact us so that we can better advise you on how to use them for your desired experiment. In conclusion, virion-display is a versatile tool that can be used to study any type of transmembrane protein. We've done it thus far with single-pass RTKs and immune receptors. The future can hold that we'll expand this to multi-pass ion channels or other types of receptors. Virion-display sets itself apart as it preserves its native conformation and post-translational modifications to facilitate testing of biologics. It avoids the need to develop unique purification schemes for each GPCR, facilitates non-biased binding since it is not a functional readout. We are just looking at unbiased binding events to the GPCRs expressed, and we need not worry about any contaminating interactions with the intracellular side. We are only limiting ourselves to the extracellular portion of the GPCR. And that these virions can act as probes to identify previously undiscovered ligands and receptors for interested animals.